Hello, I'm Vinicius Borges, and I'm going to present the work entitled Pathway Analysis of Genome-wide Linkage Analysis Regions on Essential Hypertension in African-derived Brazilian Quilombo Populations in Vale do Ribeira, São Paulo. This work results from the collaboration between researchers from Universidade de São Paulo in Brazil, University of Washington, and Marshall University. Although there are other types of hypertension in the literature, here I'll be talking about essential arterial hypertension, the most common hypertension type caused by chronic high blood pressure with pathological character. Hypertension is frequently observed with functional and structural consequences that affect heart, brain, kidneys, and blood vessels. According to the international guidelines, we classify hypertension when systolic blood pressure is equal to or greater than 140 mmHg and/or when diastolic blood pressure is equal to or greater than 90 mmHg. Hypertension is the leading cause of death worldwide, causing 9.4 million deaths per year. Epidemiological studies present projections for 2025 in which the incidence will reach 1 billion people affected globally. Hypertension is known to be multifactorial, being related to age, gender, and environmental factors, and largely genetics. Such complexity is revealed by several candidate genes and susceptibility loss, as well as unequal prevalence in populations, mostly affecting people of African ancestry. At the same time we know about the impact of hypertension, we also know that it lacks studies with admixed samples. Until recently, almost all studies are based only in European samples. Studies such as candidate genes-based, genome-wide linkage analysis, and association studies have been used to search for genetic factors that contribute to essential hypertension, but part of their heritability is not explained yet. Quilombos are Brazilian communities with a traditionally rural way of life dependent on agriculture and subsistence hunting. The communities were formed after the end of the slavery period in Brazil by individuals of African descent, constituting quilombola communities. Today, these communities are remnant of the quilombos. There are several quilombola communities in Brazil, and 8 of them participated in this work. Those are located in the southeastern region of Brazil in the state of São Paulo. They present a true hybrid ancestry, whereas the African component represents the highest ancestry proportions. These populations constitute an ideal sample to study complex disease, such as essential hypertension, because they present high hypertension prevalence, reaching 41%, and because they are semi-isolated, which helps to reduce population stratification. Also, the greater sharing of genetic and environmental characteristics amplifies the signal-noise ratio. Here, our goal is to perform pathway analysis of mapped regions of interest from linkage analysis adjusted for presence of admixture to explain essential hypertension in African-derived admixed Quilombo populations from Vale do Ribeira region in São Paulo, Brazil. By achieving this, it may help to improve our understanding of essential hypertension etiology. To achieve our goal, we constructed 6 pedigrees using GenoPro and Morgan softwares, genotyped more than 400 samples using Thermo Fisher Axon genome-wide human oranges 1 SNP array, pruned data and selected marker subpanels using Plink, King, and PBEP softwares, infrared haplotype, estimated local ancestry, and estimated allele frequency using Shapeit, RF Mix, and AdmixFrac softwares. We also computed inheritance vectors and performed genome-wide linkage analysis and then dense linkage analysis using Morgan. Our last steps were search for genes in each region of interest using Varalect, Biomart, GeneCards, and GWAS catalog to finally classify molecular and biological process and construct an interaction network using Painter, String, Cytoscape, and Clugo and Clupedia softwares. As results, 22 regions of interest were identified and confirmed between dense linkage analysis strip subpanels, as you can see in the figure on the right, And for all these 22 regions of interest, the loss score threshold was greater than 1.45 and it ranged between 1.45 to 303. 61 essential hypertension genes were found within these 22 regions of interest, as you can see in the figure on the right. This is the gene set used in the pathway analysis. We also were able to cluster the biological process in which the gene products of these genes are involved and identify that the main clusters are regulation of blood vessel, cardiac muscle tissue growth, and regulation of renal sodium excretion. By identifying and ranking the outcome, we identified that our 61 gene products are most involved in arterial, smooth muscle cell, vascular smooth muscle cell, smooth muscle, artery, blood vessel, and vascular system as tissue expression. The main disease gene associated from our 61 gene products are arteriosclerosis, C1 inhibitor deficiency, molecular weight kininogen deficiency, angioedema, ischemia, and coronary artery disease. Lastly, our 61 gene products' main biological processes are artery smooth muscle contraction, positive regulation of vascular permeability, negative regulation of cardiac muscle cell proliferation, and positive regulation of heart rate. By the end, we used all the information resulting from the pathway analysis to identify 5 extra genes related to the pathways. These genes have the potential to be related to essential hypertension, and they will be investigated in the future. Thank you for your attention. We can be reached via email or website available through the QR code.